All right, let's get started. Can you hear me in the back row? Can you hear me in the back row there? Excellent. Okay. We didn't quite get to the end of the first handout. I will just sort of skip to the very end of it. There's not a lot more that we didn't get to. Just a reminder that Monday is the final exam. I understand that some students have been asking whether this year's final will be like last year's final, which was different from the previous year's final, in that last year's final was compiled entirely out of questions from previous tests. That is an inappropriate question, and neither I, nor Angie, nor Susan will answer it, and I hope none of you will ask it again. So, now that you know how to make me tick off. So, all right. Let's just close off the very end then of the review A. We were talking about protein structure and, Sarah, yes?
I think there was a correction on the slide. Is that supposed to say it's
Where am I saying? You're right. It would be sigmoidal. You're right. Well, thank you for saying that. Good thing we looked at that. Let me see. Sigmoidal. Good thing we came back to it. Sigmoidal. I'll make that a bold. Right. Thank you. Is there anything else that people have noticed along the slides that I need to correct? Okay. One of the key things about hemoglobin is that it is a cooperative protein in which the first ligand binds differently than the last one, and like many cooperative proteins, it has a sigmoidal, as you point out, kinetics or sigmoidal binding curve. Its overall affinity is not as great as myoglobin, so it can dump off to myoglobin in the tissues. And the change from the unoccupied to the occupied involves structural changes where the subunits rearrange a little bit. So although the tertiary structures of myoglobin and hemoglobin are very nearly identical, only hemoglobin has a quaternary structure, which is why it can have cooperative behavior, can have a sigmoidal curve, and it in fact has two slightly different quaternary structures, very closely related, principally different in the size of this pocket here in the middle. And let's see if I can page down here from there. No, okay, let's go into this then. And so, let's see if we can get the, we move down, let's see. Oh, there we go, okay. I'm not going to go into a lot of detail on all of these slides because I do want to get to the second review as well. These are here for your help. But bisphosphoglycerate is this highly charged, negatively charged molecule that can fit into that hole in the middle of deoxyhemoglobin. And when it fits in there, it kind of acts like a doorstop to keep the thing open. And so bisphosphoglycerate, or BPG, is the modulator that can change the affinity of the hemoglobin by simply physically jamming the molecule as a whole in the deoxy state to an extent. It's not irreversible, obviously, or we'd all drop dead. But that's where the BPG can bind in this cavity that's created by the fact that four subunits are packing together. The BPG pocket only exists in the quaternary structure of hemoglobin. Okay, so let's close that one off then and go to review B. Yes, I want to say it. Okay. All right, so here we go. We can start up again here. This is the second of the three review packets we'll be working from. I need to do my pointer again. Arrow options. There we go. Okay. And I pointed out how BPG has negative charges. If you look at the interior of that BPG pocket, there are side chains that have positive charges, like lysine and histidine and so on. And because it makes the deoxy state more stable, it helps hemoglobin release oxygen into the tissues. The exact affinity of hemoglobin can both be tuned by the concentration of BPG. So here they're saturating it with BPG. You can imagine having a curve somewhat intermediate by lowering the concentration of BPG, which can be done, for instance, when you adapt to high altitude. Another way of tuning oxygen affinity is by using a different hemoglobin. So the fetus that it's carrying within the mother has a slightly different affinity for oxygen, so it can rip oxygen away from the mom's circulation. Okay. Let's talk a little bit about the cytoskeleton. It's another protein system, if you like. The hemoglobin and myoglobin pair are the original paradigm for proteins, and there are transport proteins, storage proteins. The cytoskeleton is a different sort of universe of protein behavior. It's something that's going to provide structure and cell type specificity for cells. And there are three major filament systems. You have microfilaments, which are going to be the hallmark, if you like, of things like muscle cells, where they're first isolated. Platelets that can contract and make a clot get small. Microvilli that stick into your gut. Second system of microtubules that are like little rigid rods that give the cell a different set of capacities. You're going to see those in the cilia that can beat and drive water through your lungs, unless you're a smoker, of course. Make neurons long and skinny. Not joking about that. You don't want to see smokers' cilia. They're not pretty. Third major family are the intermediate filaments that are there for strength, that hold the skin together, that hold other epithelia together. So we see them in skin. Our hair is essentially pure intermediate filaments that have been cross-linked. Epithelia are the lining tissues that line body cavities and so forth. They're all going to have intermediate filaments. But there are also special ones for neurons, because these long, skinny cells need strength, and the intermediate filaments help provide strength. We just mentioned that about the intermediate filaments. The major features for microfilaments are to hold on to things, for adhesion, cell-cell adhesion, cell-substrate adhesion, contraction, so the work of muscles is to contract, and to provide stiffness. There are places where they do that. They are built up out of a protein called actin, which can be found as a free monomeric protein. Their filament length is fairly small. You don't need to memorize 7 nanometers, but they're microfilaments because they're the small filaments. Microtubules are another sort of family of filaments. We talked about them briefly in the previous slide. Here we've highlighted them in green. Some of the cells, like ciliary neurons. In the cilia, they allow either to push the water past them or for cells to swim. So microtubules give sperm their motility. They provide for stiffness in the inside of the ear. And in every cell that goes through cell division, they're needed for mitosis. So kind of essential to life. That role in mitosis is why anti-microtubule drugs are also anti-cancer drugs. Microtubules need 2 proteins to be built. They have an alpha and a beta tubulin, and there's 1 alpha and 1 beta in the dimer. So that's the building block for microtubules. Intermediate filaments, they're hollow tubes. They are actual tubules. Intermediate filaments, we've mentioned where we find them. They really provide for strength of various kinds. They are abundant in the tissues that have to resist mechanical distortions, and they are always present when there's major attachments to either cells or to tissues. They are much more various in the protein composition. So there aren't merely 1 or 2. There are 5 families of intermediate filament proteins, one of which has 40 different members. So a lot more variation in what the proteins are that make up intermediate filaments. Let's see, one more. Here we go. If we look at the biochemistry behind these structures a little bit, here in the center you've got your actin filaments, actin monomers. They have to polymerize to make those filaments, and they do that in an asymmetric way, so that these are going to have distinct ends. The top and bottom ends are different from each other. So you can talk about a plus and a minus end in terms of assembly and so on. In other words, microfilaments are polar filaments. They have ends that are different from each other, and it takes ATP to make them form. Microtubules are also polar filaments. They have a fast and a slow end. They take GTP to form. By contrast, intermediate filaments are bundles of protofilaments, and they are anti-parallel packing in them, so the filament as a whole is symmetrical. They're passive. They don't need any kind of nucleotides. They form and they basically stay for the most part, although a few of them cycle during mitosis. Intermediate filaments was where we first met a structure that we've met in a number of other places as well, which is sometimes has been called a supersecondary structure. But it's a place where the secondary structure of an alpha helix is wrapped around another alpha helix. So you can see that here. Here's one alpha helix. There's another one. And because they have a repeating pattern of hydrophobic residues, those hydrophobic residues stick on each other kind of like a zipper closing up. And it's also called a coiled coil. It's a heptad repeat because every seventh residue is going to have that hydrophobic character. The formal name is a coiled coil. And I mentioned the hydrophobic interaction. Intermediate filaments do not need nucleotides to do this. And as I mentioned, this approach to making dimers is not unique to intermediate filaments. We'll see it many places. Okay. Okay. So that was a whole sort of superfamily or a tribe of structural proteins whose principal role is structure. They do break down nucleotides to do that, but their role is basically structure. We need also to think about proteins that can actually catalyze things, or enzymes. And so we looked in a little detail at one particular enzyme, the chymotrypsin, which has been a model for many, many studies of enzyme activity. It's a hydrolytic enzyme, can just take that peptide bond, add water across it, break it into two, and that's found in the stomach in order to break down our protein. Pretty fast here, 190 bonds per second. That'd be the turnover number for it. 10 to the 11th times faster than without. That is, we can find proteins from mammoths that are tens of thousands of years old in the presence of chymotrypsin. We're talking about seconds. Now, if you look at the protein as a whole, it has a tertiary structure, as proteins in general do. And if you look at it a little more closely, you see there's like a cleft, a hollow space, which is where the actual event of hydrolysis will happen. And we call that the active site. Every enzyme has an active site, someplace where something interesting happens. In the case of chymotrypsin, that active site is formed between two domains. So each half of that enzyme kind of folds to itself to a certain extent. It's fairly specific, not very nasty. When we want to think about enzyme reactions, it's useful to have a way to sort of show the progress of the reaction so we can think about the different aspects of it. And we use for that something called a reaction coordinate. It has a vertical axis that has a real number attached to it, which is free energy, or G. The bottom axis is sort of how far you've gone through the reaction. It's not time or anything like that. It's just sort of how far have you gotten down through the reaction. If you start with reactants that have a certain amount of free energy, and you have products that have another amount of free energy, if the products have less free energy, if they have a negative delta G compared to the reactants, you're going to have a spontaneous reaction. If the products have more free energy than the reactants, that reaction will not go spontaneously. Energy will have to be put into that reaction somehow. You remember we mentioned on Monday that it can do it by coupling it to a spontaneous reaction, but by itself it won't go. So we know if there's a negative delta G, it's not a spontaneous reaction. If there is a negative delta G, it tells you whether it is spontaneous in the sense of whether energy has to be put into the reaction. It does not tell you about the rate. It might still take a very, 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 very long time, because the delta G of the reaction is not the thing that determines how fast it'll go. It's whether it goes to one side or the other. And what's going to determine how fast a reaction goes is the height of the transition intermediate. So you notice there's a hill in each case between the reactants and the product. This is the transition state. This is a place where you have to get, from A to B, you have to go through somewhere else. And the somewhere else is the transition state. It's a configuration, a strain shape, whatever, that is neither the reactant nor the product, but it's what you go through along the way. And it's always going to have a higher energy associated with it than either the reactants or the products. And I think it should be intuitive that if you have a big hill to go over, it's not going to happen as often. If you stand here and you throw marbles up a hill, if it's a small hill to go off and fairly over, if it's a big hill, you're going to have to do it more often before you're going to happen to get enough energy to do it. And this is the magic of a catalyst. It's not just an enzyme. Enzyme is a protein catalyst. The magic of a catalyst is it somehow or other reduces the transition state's free energy change. It makes that hill shorter. There's something about the catalyst that allows the reactant to get to the product through a transition state that involves a lower free energy change than in the uncatalyzed reaction. So it changes the rate of the reaction because it changes how much energy it takes to get to the transition state. Catalysts cannot change the delta G of the reaction overall. They can't do that. So catalysts can't change the equilibrium values, if you like, of the completed reaction. All they can do is change the rate at which it happens. Any questions about that before we go on? Okay. All right. So enzymes are specifically protein catalysts, and catalysts somehow or other change the transition state's free energy. How do they do that? They do it by creating some kind of chemical condition, some chemical environment that alters that free energy change for the transition state. Some of them are going to need cofactors to do that. That's why you should get your vitamins. Most of the B vitamins are cofactors to enzymes. And we see certain mechanisms come up over and over again. So 3 very common mechanisms involve either an acid-base catalysis. So the protein is acting like a very specific local environment to catalyze a particular acid-base reaction as opposed to your test tube in the chemistry class, which is gross and nonspecific. Some of the reactions are covalent, where the reactant starts and actually then becomes covalently attached to the enzyme itself, and that either preserves energy in some way or allows a state to be reached that can't be reached any other way. And the third common one are metal ions, that the metal ion is sort of sitting there in the enzyme and allows chemistry to happen that can't happen from just the side chains of the amino acids. And chymotrypsin was kind of a nice enzyme for people to start studying in the early days because it actually is an example of 2 kinds of catalysis. It uses acid-base catalysis, and it also uses covalent catalysis. And it does that through this very cute little catalytic triad, aspartate, histidine, and serine. These 3 residues are present at the active site. They're the catalytic residues, and the specific geometry of those catalytic residues is what accomplishes the specific addition of water at just that site. So the catalytic residues are an important part of the active site. So what is the rest of the enzyme for? If it takes 3 specific residues to make chymotrypsin an enzyme, what's the rest of that stuff for? This is some of the ways of thinking about, for instance, the early ways of thinking about an enzyme was like a lock and a key, that there was a shape that you fit it into. And that model was kind of good for thinking about specificity, because enzymes are very specific. But it wasn't as good a way of thinking about getting things back out again, because being too tight is as bad as not being there at all. And this is an example of how you could have a worse rate if you bind too tightly. So here you have your substrates, here you have your products. If you're binding too tightly to the enzyme as substrates, you've actually increased the free energy change to get to your transition state. So that model is not perfect. It was a good beginning point. Some of the things that we now understand contribute extensively to how enzymes pull this trick off. Just proximity, how close they hold reactants. Orientation, twisting it a little bit like this so they're lined up right. Sometimes the enzyme will actually change its shape when the substrate binds and then can kind of grab it some new way. The electrostatic cloud, in other words, positive and negative charges around the substrate that can influence where the electron clouds go. Another nice thing it can do is to keep water out. Many of these reactions have water as either a competing reactant or as something that could be needed in a different step and so forth. And so part of what the enzyme is just doing is making sure that not only are the two reactants close to each other, but that water isn't, because it could get in there and compete too easily for things. And this is just a little diagram showing you proximity and orientation. Another nice thing about the proteases is they show us some things about protein structure and about enzyme evolution. These three enzymes, chymotrypsin, trypsin, elastase, all have very similar tertiary structures. You can see them overlying here, very similar, almost as similar to each other as myoglobin and hemoglobin. However, their primary sequences are quite different. Just as in the case of myoglobin and hemoglobin, primary sequences were quite different. And so one of the things this tells us is that there's many ways to arrive at a particular tertiary structure. And we also see clues from the differences as to how they got there. Even though the primary sequences have changed so much, these three residues, the catalytic residue, the asp-His-Ser, in the middle of that active site, those are completely conserved. So a lot of the primary structure, the sequence, varies. You know, you can have any old thing you like on the surface. It could be a serine, a threonine, a spartate, whatever, as long as it's hydrophilic. You can have a leucine or an isoleucine in the interior. You can have an alanine or a leucine, whatever. But you have to preserve those catalytic residues because they're the ones that are going to accomplish those tricks of proximity, electrostatic, and so forth. The surfaces which are interacting with water are the place where the greatest variation of residues is going to be. So we've talked a little bit about the catalytic residues at the active site. They actually are involved in changing the height of that free energy change for the intermediate. But the other thing about enzymes is that they're specific, that they can make one thing react but not another one. And these give us sort of a clue to that. You don't need to remember which ones do which, but the fact that they do things as different as hydrophobic things or positively charged ones or small ones is accomplished by the fact that in addition to the catalytic residues, which are actually doing the catalysis part, if you like, there is a substrate pocket that confers specificity. The residues in there will pick out some things, not other things, so that the catalytic residues will only act on that particular substrate. So kind of a cooperation, catalytic residues doing actual catalysis, substrate binding pocket choosing who gets to be the target, holding on to the substrate while it happens. Little taste of kinetics. In this graph, we're looking at how fast a reaction is going. And if you add more substrate to a constant amount of enzyme, you'll get more and more product being formed at a higher rate of formation up to a certain point. So we can say that a given enzyme has a maximal velocity that it can catalyze a reaction at. And so when an enzyme is completely saturated, it's going to go at Vmax. This is a hyperbolic curve for reactions, just like myoglobin has a hyperbolic binding curve. You don't actually get to Vmax, but you can approach it as you, depending on the solubility of your substrate. If you're holding the substrate constant, you can vary enzyme concentration and see that you can get a faster reaction by adding more enzyme to it. So if you have saturating amounts, you can then increase it that way. So it would be nice to have a way to talk about these things, and I'm not going to go through the exact equation here or the derivation of it, but sort of looking at the landscape here, there are two important features of enzyme kinetics that we can look at. And one is the Vmax, how fast that enzyme in principle can go if it was completely saturated with substrate. And that's how fast it turns over when there is substrate bound. And a second number is the Km, the Michaelis constant, which is, in practical terms, going to be observed when you've gotten to half of the maximal velocity. That's the concentration of substrate it takes to get to the half maximal velocity. Just as we looked at the half saturation of hemoglobin and myoglobin to compare their affinities, we use the half maximal concentration to look at that. So the Km is a measure of affinity, of how readily that enzyme binds a substrate. Vmax is a measure of how quickly it can turn it over when it's occupied. Km and Vmax are completely independent properties. They are completely independent properties. One way to think about it is that the Vmax is a reflection of those catalytic residues, the asp-His-Seer, and how good they are at twiddling their fingers. And that the Km is, to some extent, a measure of how that substrate specificity pocket finds and binds and holds on to the substrate. But they are not connected properties per se. They are separate properties of an enzyme. Any questions about that before I go on? Okay. Going through those derivations, you can arrive at the math behind that. If you start looking at things that are simpler, things that are more complicated than A goes to B, the math gets much more complicated. And so if you look at something like this, where you're having fructose 6-phosphate bind to a transkylase, and it can take it apart into 2 and 4 carbon pieces and put them together in different ways, this is obviously a much more complicated reaction. And every one of those arrows, every single arrow in that diagram has a rate constant associated with it. But you can still speak of a kcat for the reaction overall. Now, there are some enzymes that are more complicated than that. Just as the binding behavior of hemoglobin was more complicated than myoglobin, some enzymes get a little fancier than just simply A goes to B. And we can see that in their reaction curves because they have a sigmoidal shape to the curve instead of the simple hyperbolic shape. Now, just as the sigmoidal shape in hemoglobin was a sign to us that there was probably cooperative behavior going on, again, in general, enzymes that show sigmoidal curves have cooperative behavior and are almost always multi-subunit enzymes. We call the fact that you can have binding on one part of the enzyme have an effect on the other reaction sites, the other catalytic sites, allostery. Allostery. Allo is other, steer is steric place, site. So allosteric effects are when binding at one place on the complex alters behavior somewhere else. Hemoglobin is an allosteric oxygen transporter. Even though the math gets fancier, allosteric enzymes still can be said to have a Vmax, that is the maximum velocity which it would go if it ever got fully saturated. And they can still be spoken of as having a Km, the concentration that half saturates it, and therefore is a measure of affinity. Km is still pretty much a measure of affinity. Vmax is still a measure of turnover when completely occupied. Enzymes don't always go. Sometimes they get inhibited. And sometimes they get inhibited because we want to inhibit them, and sometimes they get inhibited as part of metabolism. And there are a number of kinds of reactions that can go on. One form of enzyme inhibition is irreversible. So, for example, if you took chymotrypsin, we were just talking about it, and put a substrate on it that reacts and then becomes covalently cross-linked to it, it's dead. It's irreversibly inhibited. We sometimes call that a suicide substrate because the substrate dies in the process. This is an inhibitor we made up to study chymotrypsin, But in our bloodstream, there are irreversible inhibitors that are proteins that are willing to die to inhibit proteases and stop the process of clotting. They're suicide substrates. A second kind of inhibition is competitive inhibition, and this is reversible. It's one of the most common kinds of inhibition to find. And the idea here is that the substrate that should bind to it and the inhibitor that's getting in the way both have features, in this case the little pointy ends, that map to part of the active site. They map to the part that's the substrate binding site generally, but they are not able to be targets for catalysis. So they don't interact with the catalytic residues the way the real substrate would. So it can bind at the active site, but it doesn't undergo reactions. It doesn't undergo catalysis. So that's the conceptual idea behind a competitive inhibitor. They're competing for that active site. If we look at what that looks like in kinetic terms, here we have, I'm sorry about the red-green. I've looked for these graphs without red-green. Sorry about that. Here in the upper line, the green line, you have the enzyme reaction behavior in the absence of the inhibitor. So there is some substrate that gets, concentration of substrate that gets you to half maximal. This is its affinity. It's Km, a measure of its affinity, more or less. And it has some rate at which it can go when it's fully saturated. It has a Vmax. If you think about it, it should make sense that a competitive inhibitor changes the apparent Km, the apparent affinity for that enzyme, because a significant fraction of the time it's the inhibitor that's there instead of the substrate. So you have to raise the concentration of substrate before it starts to win the race. But if you get it occupied with substrate, it still reacts at the same turnover number. So a competitive inhibitor can change the Km, it can decrease the affinity if you like, it can raise the Km, it raises the concentration it takes to get to half maximal velocity, but does not change the catalytic residues, and so they can turn over once the real substrate gets in there. Questions about that? Allosteric inhibition is an example of when you can have typically an actual metabolite come back and bind to typically an enzyme that has complex sigmoidal behavior. And so it is binding somewhere on the substrate, on the enzyme, and changing other sites. Feedback inhibition is one of the common examples of that. You can also have allosteric stimulation, where it turns things on instead of turning them off. Competitive inhibition only turns things off. But allosteric regulators can be inhibitors or stimulators. Here's an example of a very important enzyme that is controlled by a variety of allosteric effectors, allosteric binding things. Phosphofructokinase here. And this is one of the, here's imprinted down there, phosphofructokinase. Phosphofructokinase is right in the middle of glycolysis, and it is the key control point for glycolysis. It turns the rate of glycolysis up and down. And phosphoenolpyruvate, of course, is one of the nearly the end products, not quite the end of glycolysis. And it can alter the behavior of phosphofructokinase in a very interesting way. Here is the behavior of phosphofructokinase in the absence of this nearly end product. It's got hyperbolic kinetics. And here's the behavior in the presence of phosphoenolpyruvate. Unchanged in terms of the Vmax. Big difference in the Km, or the affinity. And so in the concentrations of substrate that the cell sees, this would translate into a decrease in activity. So we've talked about cytoskeleton proteins, we've talked about enzymes, we've talked about nucleic acids. The other big domain of structural proteins would be membranes. And these are a place where lipids come into their own. They are the boundaries that allow cells to have insides and outsides. The things that go into membranes are mixtures of lipids and then membrane proteins, and they are bilayers. The bilayer aspect controls movement in a gross way, keeping hydrophilic stuff out, and then the proteins add function. In contrast to the nucleic acids or the proteins, membranes are not covalently put together, but they do form cooperatively. I talked a little bit about the fluid-solid mosaic model of membranes, about how the lipids are pretty fluid, but the proteins don't move a whole lot. They behave more like a solid mosaic. And that membranes allow cells to have electrical status different than the outside. Here you can see that in a little more detail. You have things like integral membrane proteins that can let molecules get through, or they can be hormone receptors, peripheral membrane proteins, and everything that's sort of at the level of the head group out is going to probably be hydrophilic, and everything here in the middle of the bilayer is going to be hydrophobic. This is just showing you those components again in a little more detail. That the lipids, because they are not attached to each other for the most part, the lipids can move pretty much with diffusion. But the proteins are, in general, attached to the cytoskeleton. We just talked about it. And their movement side to side is much slower. Both of that kind of motion that I've talked about so far is in the plane of the membrane, side to side, like that. Flipping from one leaflet to the other leaflet is not very common. And that means that membranes can be different on the two sides. They can have different properties on the inside and on the outside. To get things across membranes, well, hydrophobic stuff, if it's a little bit soluble in water, can just float through the interior. That's not a very common thing, but it's how steroid hormones, for instance, get through. There is some that's passive, like the acetylcholine receptor or channels, where you create a hole and things can just flow through. You can have active transport, where you actually invest ATP and pump things up against a gradient. And ATP is the most common thing that's used to power a pump that actually moves things out. Another way of transporting things is to couple the passive transport to a gradient you created with the active transport. So for instance, you can use a pump to create a sodium gradient, and then use the sodium gradient to pull glucose in through a co-transport process, or a SIM port. The membrane channels are selective, so a sodium channel won't carry potassium through it. And they're very fast, many, many molecules. Gap junctions are a third kind of membrane contact that lets cells talk to cells and communicate chemically. This is going through that again. This is just sort of your diagram of a channel. You're going to either be polar or ionized, something hydrophilic there. Examples of the passive diffusion would be the steroids. And there's a picture for you of gap junctions, which are involved in letting cells talk to cells chemically. I think we're getting a little bit behind. So here we're going to the other sort of not terrifically abundant members, but very important members of the membrane, which are membrane receptors. And they're going to have, the membrane receptors have a fairly common strategy of doing that. So you have your signal, which is a chemical in most cases, that can be bound to surface membrane. Some kind of a process of amplification, so that one molecule at the surface generates many molecules on the inside, and those amplified molecules have to then in some way trigger the response you want to actually get. A common form of that transduction is protein phosphorylation. There's an enzyme that can recognize its target and do that. So let me just see how far we have to go. And I think I may want to let you look at these yourself. I think we still have. I am going to suggest that you look at these things about the receptors on your own, because I would like to get to metabolism today. And I think those are fairly straightforward. Metabolism is the whole constellation of stuff of either getting stuff out of the environment or building stuff up. And I'm sort of provoking your memory here to sort of think for yourself of which of those pathways that you know of are connected to each other. They're driven in most cases by burning carbon fuels, oxidizing them. And that oxidation may be with oxygen and may be without oxygen. Glycolysis is oxidizing carbon but does not require the presence of oxygen. And these pathways often reuse approaches to things from one to another. When we look at taking fuels and oxidizing them to carbon dioxide with energy being released, that is catabolism. Breaking things down is catabolism. So glycolysis is a catabolic pathway. Oxidative phosphorylation is a catabolic pathway. You might want to prod your memory and think of what other catabolic pathways you know. If you have energy in a usable form and small molecules to build up bigger ones, that's anabolism, like anabolic steroids. Gluconeogenesis, you're building up glucose molecules. Glycogen synthesis, you're building up glucose polymers, glycogen. Photosynthesis, you're building up sugar molecules from carbon dioxide. We've talked about a few other anabolic pathways. And then a few pathways do both. They can both build things up and break them down, depending on the needs of the cell. Two of the preeminent ones would be the citric acid cycle, that sort of hub of metabolism, and the pentose phosphate shunt that can also be used either to make building blocks or oxidize carbons. We've talked about another one as well that's amphibolic. You've come to recognize the compounds like ATP that have a high energy potential transfer. And this is just sort of a reminder to you of some of the other compounds that have an equivalently high energy bond, speaking loosely. There are carriers for almost everything. You can carry carbon fragments. CoA is the common carrier for those. You can carry electrons. These are all electron carriers. NADH is the carrier for oxidation, for catabolism, NADPH for synthesis. Quinones we've seen both in oxidative phosphorylation and photosynthesis. Cytochrome C, ferredoxin. You can carry sugars, commonly with a UDP. So all of these little pieces typically have a carrier that both makes them specific and in some sense activates them for the reactions they need to be used for. And this is the grand overview then of all of metabolism, of the polymers that can get broken down to specific monomers. They feed all to a common entry point into the citric acid cycle of acetyl-CoA. Proteins can also go to a few other things as well, but certainly all three of those will end up at acetyl-CoA. The citric acid cycle, in the process of burning it off as carbon dioxide, is also going to be producing NADHs for the oxidative phosphorylation. And then oxidative phosphorylation is the common end pathway for burning any of these fuels, and it's the most energetically efficient in the sense that it produces the most ATP. If you just take glucose down to this point here through glycolysis, a little bit of ATP. Take it clear down to oxygen as the electron acceptor, much more. And the next few slides are sort of side-by-side comparisons of some pathways that we've talked about to let you kind of refresh your memories about their properties. And some of them have names that are similar to each other, and so it's nice to keep them straight. So glycolysis and gluconeogenesis are the catabolic and the anabolic pathways, the catabolic and the anabolic pathways for glucose and for some other sugars. They're both going to be using 3 and 6 carbon phosphosugars. They both are controlled one way or another. They're both in the cytoplasm of cells. Glycolysis happens in all tissues, and it's there to release ATP and acetyl-CoA. And fructose 2,6-bisphosphate, which is this trace amount made by phosphofructokinase 2. Remember, it's a little tricky, different enzyme there, activates it. By comparison, gluconeogenesis is an anabolic one. It's only going to happen if you've got abundant energy available. Only the liver and the kidney can do it. and it costs energy, both ATP and GTP. Fructose 2,6-bisphosphate inhibits that one because you don't want to be wasting energy at the same time. And finally, the reactions of the two are very similar except for a few bypass reactions at the irreversible steps, and that's going to cost energy. To get uphill at an irreversible step, you have to invest energy. So it's going to cost ATP and GTP to make sugar out of carbon backbones. Two of the major cycles we've talked about are the citric acid cycle and the Calvin cycle. They're both inside of an organelle. They're both controlled principally by allosteric controls. Citric acid cycle is in the mitochondrial matrix. It is an amphibolic pathway. All tissues have it in us, and almost all eukaryotes have it. I think there's a handful of parasites that don't have mitochondria. And one of the points of the citric acid cycle is to release NADH for further energy production and oxidative phosphorylation. But it's amphibolic. It's also going to produce backbones that can be used for fatty acid synthesis or amino acid synthesis, gluconeogenesis. So that's the amphibolic character to it. Calvin cycle, by contrast, is in the chloroplasts, so therefore only in the leaves. And it's going to use NADPH, the anabolic form of an electron carrier. It has some overlap with gluconeogenesis. So there's some places there where, well, you're making sugar. You shouldn't be too surprised they have some similar reactions. Which gets us to the organelles themselves, that the chloroplasts and the mitochondria are these double membrane-bound organelles inside of cells. They both use this trick of passing electrons from one transmembrane protein to another inside of a membrane to generate a proton gradient. Very sneaky way to make ATP. So you make a proton gradient, and then you have a special sort of water wheel that carries the protons back, and that's what's used to generate ATP. For chloroplasts, those electrons come from water, so you make oxygen. Whereas in the case of mitochondria, the electrons are coming from the fuels, from fat or sugar or whatever, and it's going to use up oxygen. Glycogen is not quite as universal a compound. It's important to us, not to trees. Those two pathways are only going to be in a few tissues, muscle, liver, kidney. They have dual control, local as well as hormonal. But these are two of the pathways that we've talked about that really have a place for hormonal control. So epinephrine is going to inhibit the pathway. Insulin will stimulate it because you have extra sugar around. Insulin's a marker for extra sugar. Make some glycogen. We've got time to do that. Whereas the opposite set of reactions in terms of glycogen degradation. Epinephrine is the sign that there's a tiger on your back. You need to release your sugar so you break down your glycogen. And I think that is 20 after. I'm happy to go on if people want, but if you want to step out, that's fine too. Sure.
What does that stand for?
Compounds.
Compounds. Oh, okay.
Yeah.
All right.
Is there anybody who wants to stay for the last few slides or not? I'm happy to stay, but don't feel like you have to. Hands up if you want me to stay. Okay.
Write stuff in the review packets, or?
Well, I think start with the
Small hole of zero.
Hey guys.